Hi, this is Miss Litton with a little bit of a scratchy voice. This is my excellent AP biologist, 7th period. Say hi. Hi. All right, and we are going to review chapter 12. These are just phenom students because their quiz is not tomorrow. When is your quiz over chapter 12? Thursday. But yet they are dedicated and come in, so I really appreciate your maturity. All right, so as I said before, here are the 6 now. Big ticket items related to this chapter that I want you to feel comfortable with. Any of these could be turned into essay questions, right? So these are the 6 things. You want to maybe take note of it, and then, okay, there you go, paparazzi. And then make sure you understand that upon the close of our review. And if not, I believe I have an open scheduled for Wednesday, right? So then you can come in and get some one-on-one help if you want to over any of these topics. So the first thing is that I want to talk to you about is just the conundrum of how could DNA be our actual hereditary material? And that was an issue because DNA only has a variation of what? 4. What's the variation of 4 that it has? I'm sorry, I cannot hear you. Okay, and what are those? Bases, right? So DNA is made out of nucleotides, and DNA only has a variety of 4 bases in their nucleotides, whereas proteins, it sounds so much more likely it has a variety of what? 20, because there's 20 different R groups. So there is a series of experiments just used to identify the DNA as our actual hereditary material. Remember, there were 3 things they knew it had to be able to do. Do you remember what those 3 things are? Turn towards your bio buddy and see if they can remember what those are. Okay? S squared M. S squared M, right? What are the store information? It had to be stable, but it also had to be able to, which sounds contradictory, right, to the stability part, right? But it's not. It just means that it can change. It can vary. So our first experiment that we would say, what was our first experiment? Griffith. How are we going to remember Griffith? Griffith Observatory. Griffith Observatory probably has a lot of what? Rats and mice. Good. So Griffith just used the mice to tell us about the bacteria. So there were 2 strains of bacteria: S strain, R strain. S strain was smooth. It was it would kill the mice that it was injected into, whereas the R strain, the mice would live. So that was repeated again and again and again. Okay. But then what he would do is he would take the S strain and he would heat it up, the bacteria in a test tube, heat it up, inject it into the mice, and the mice would what? Live. Because something happened to the ability of those S strain to survive. They didn't have any metabolic pathways.
Colin Welch, please come to the counseling office. Colin Welch, please come to the counseling office.
Oh, Colin. Now, so they were definitely dead. So then they took some dead S and mixed it in with some what? Live R. And lo and behold, the mice start dying again. So what he proposed is that R picked up something from the dead S. Now, we call that today transformation. That R got transformed from something from S. All it had to get was the code, right? It had to get whatever made the S lethal. It had to acquire that. We know now that the genetic code is the DNA, so it picked up some of the fragments of DNA from dead S, put it into its DNA, and made it then lethal. Okay? So then we have the law firm. Who's the law firm? They're not really a law firm. We're just using them to remember. What are they? Avery, McLeod, and McCarty, right? Those 3. So those took it a step further to find out what was the stuff in there that was actually doing the transforming. So they used enzymes. Enzymes that would degrade proteins, in case that was the transforming something, or degrade RNA, if that was the genetic code. And when you degraded the proteins in the RNA, you still got that bacteria getting lethal. It was still becoming lethal. But when you used enzymes to degrade DNA, that R could not become lethal. Because what it was doing is it was degrading the lethal S's DNA and R's behaviors were not changed because R did not get any new genetic information. Okay? Look at your bio buddy. Do you own that? You own that material. You have. All right. Now, we talk about transformation today. When we do our genetic engineering lab, you are going to give DNA to bacteria and transform them and give them new traits. Because you're going to give them a different genotype so that they have a different what? Phenotype. You're going to alter their phenotype by altering their genotype through the process of transformation. But yet we have more experimenters, right? Who's next on our list? Hershey and Chase, because we would always chase down a Hershey bar. And they worked with the bacteriophage. Now, we know what bacteriophages look like today because we have electron microscopy. They did not. They were doing this experiment blind. They had viruses that they grew up either with radioactive phosphorus, P32, or radioactive sulfur, S35. So they were isotopes so you could trace them. Now, they knew viruses had the ability to overtake other cells and get them to make more of themselves. What did they use for the overtaking, right? What was the code they were putting in there to overtake? Was it a protein code or was it a nucleic acid code? So they labeled proteins and they labeled nucleic acids. They allowed what bacteriophages do, which they didn't know exactly. They knew they did something. And so they knew viruses had protein on them, but that protein turns out to be just the capsid that contained the nucleic acid inside. Okay, so they ended up labeling the capsid and the nucleic acid, but they didn't know that at the time. What those bacteriophages do is land like the lunar lander and then with its legs and inject the nucleic acid into the cell. And what they were injecting in there was the isotope of phosphorus, because phosphorus, right, nucleotides are made out of what? Base, sugar, phosphate. So it was injecting the isotope of phosphate, base, sugar, phosphate, the phosphorus and the phosphate, and overcoming those cells and getting them to make more viruses. They were not injecting any of the protein. And the way you know that is they did an experiment where you could have one virus with both labels for that matter if you want to. They allowed them to infect, and then once they knew they'd given enough time to infect, they put it in a really nice, what? Blender. And the blender would separate the components. Whatever wasn't injected then got spun off, right? And then they took that and put it in a centrifuge that spins. The heaviest things go to the bottom. Between virus and bacteria, who's heavier? Bacteria. So the bacteria went to the bottom of the tube, and there you saw radioactive phosphorus, where at the top of the tube, what was still with the virus itself, which had never left the virus, was the radioactive sulfur. Because it did not send protein, it sent nucleic acid. Okay? Ask your bio buddy, are you golden pony boy? Okay? So, of course, there were so many more experiments than that. Those are just the key experiments to identify that DNA is our hereditary material. Now, the light was green to figure out the structure of our hereditary material. They knew the recipe. They knew just like in a recipe you need brown sugar and you need flour and you need salt, right? They knew what was going in there, but they didn't know how it was arranged. They didn't know how it was arranged in the DNA. They knew the ingredients, but not how it's arranged. So they had things like bond angles. They knew the ratios of some things. But one of the very first experiments was a man named Chargaff. And Chargaff, when he did his experimentation, he came up with a couple of things. Do you remember what the couple of things are? Anybody want to offer? Yeah.
And no matter what species, the A's and T's are always equal, and the G's and C's are always equal?
Yes.
But,
And, go ahead.
Proportions of A's and T's versus G's and C's are different
for each species.
Yeah.
That's perfect. That's exactly what Chargaff shows. So now all of a sudden, the people trying to come up with the arrangement and how the structure of DNA are like, well, one thing I know for sure is A's equals T's and G's equals C's. Now we know that adenine is a purine now, and it has 2 rings, and it binds to thymine. And how many hydrogen bonds are between adenine and thymine? 2. We know that now. And guanine and cytosine have how many hydrogen bonds between them? 3. And we know that a purine, the number of purines equal your pyrimidines, specifically A's and T's and G's and C's. We know that now. And that's why the diameter is always the same for the DNA. Remember that? And the diameter, remember, 0.2 nanometers? 2 nanometers, sorry. And that's because a 2-ringed is always connected to a 1-ring. Perfect. Okay? Now, we know that, you only have, one of the concerns is like writing music with only 4 notes. You don't have a lot of 4 notes to write this beautiful piece of music, which is our genetic code. But it's because there's so many of them that it lends itself to its variety. Okay, so that was the other thing that was determined. So when we look, this is what you already gave. That was Chargaff's rules. Here's an example. Now you have some other experimenters. Who's next on our list?
Yeah.
Rosalind Franklin and Maurice Wilkins. Okay? And what they were doing is they were taking crystals of DNA, shooting X-rays through them, just like if you broke a leg and you shot arm here, and you shot X-rays through it, and it makes a pattern, right? They were shining X-rays through material, and it was making a pattern. And this right here showed you that there were repeating units. We don't know the spiral yet, but there was a spiral. There were repeating units, and that the diameter was consistent. That's what they could tell because they studied things like that. There were repeating units, and the diameter was consistent. So now, who comes along next? Who actually gets the prize? Watson and Crick actually get the prize, okay? And so they come up with, rearranging with the information they have from everybody. Maybe some got legally and some illegally. They come up with the double helix, okay? The double helix. And if you remember up here, oh yeah, I didn't lecture this. If you look up here, do you see 2.34 and 3.4? Do you remember what I told you about that? Okay, so DNA is number 1. Okay, and when you study the structure, it's 2 nanometers across. Okay, the space between each one of these rungs right here is 0.34, and you have 10 rungs in your ladder for a complete turn. So one complete turn would be 3.4 nanometers. It's just a little bit of trivia. I don't know, if you have to write on DNA, you might throw that out there and sound very smart. Okay, so that's easy to remember, 1, 2, 3, 4, right? Okay, now we went through already base sugar phosphate. You should feel comfortable with that. But one thing we notice here is that the sugars are going in what? 2 different directions. And this becomes really important for both replication and transcription, right? When you're trying to read the code. Because we know enzymes are very specific. And they can only read in the 3' direction. So when we look at something like this, remembering our numbering, carbon number 1 is always attached to the nitrogenous base. We cannot go on the roof because who's on the roof? Oxygen on the roof. So we can't keep counting, so we go down. So here's 2, here's 3. If we have an eyeball right here, we're looking at the 3' end, 4, and the chimney makes 5. Okay? So when you go into DNA and remember how you could label all of that? When you go into DNA replication, okay, when these go. Unlock. Go. When these bases unwind and unzip, we can't just start here at either end and just go for it and do the same thing over here. Remember, if this right here was the 3' end, then this has to be the 5. Then over here has to be the 5, and this has to be the 3. So we can't just take off of our primer. Remember, we have to wait for some of it to be exposed and then work our way backwards. Wait for some to be exposed and work our way backwards and work our way backwards. And that's why you have to have, where you only have primer put down one time for your leading strand, you have to have it put down multiple times for your lagging strand, right? And then what do we call these strands of DNA that were made in between the primer? Those are called Okazaki fragments. And who is the enzyme that reads off of this to make complementary 5-3? What's his name? DNA polymerase 3. But then we have to get rid of these primers, right? Who's the one who can rid us of the primers? DNA polymerase 1. He is the one to save us. And then he will replace that with DNA, but we need something else too. What else do we need? Ligase to seal the phosphate sugar backbone so it's continuous. And then these right here are sister chromatids held together by a centromere, and these will separate during anaphase. Click, click, click. We got that? Okay. So that's why we looked at this diagram right here. Let's do the DNA replication part first. So, here we go. Supercoiling, yeah. What is this called? Topoisomerase. So it undoes the supercoiling. Then we unwind and unzip. Helicase. Now, what held these together? When we unzipped them, what were we unzipping? That's right. And they want to zip right back up. So what holds them apart? SSB. Good. Then we have a leading and a lagging because they're going in two different directions, right? Then we need to use, thank you. RNA primase. We lay down primer. Once on the leading, primer, primer, primer on the lagging. Then we use DNA polymerase 3, and we boom, 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 right? So if there's an A, we bring in a T, G, C, G, A. Good. Boom, boom, boom. And on the lagging strand, boom, 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 boom. Now we need to get rid of the primer. We're going to use DNA polymerase 1, primer, 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 primer. And then we're going to use ligase to seal the phosphate sugar backbone. These are going to twist up. Sister chromatids held together by a centromere separate during anaphase. Okay? Ask your bio buddy, do you own it? All right. Now, when we compare and contrast, in this case, we're going to compare and contrast prokaryote and eukaryote for both replication and transcription and translation. You can compare and contrast prokaryotes for both of them. Okay? And by the way, that song is on TikTok and it's on Instagram.
Thank you.
And it's on YouTube. Okay. All right. Okay. When we have eukaryotes, we have linear chromosomes, yes? But prokaryotes have a single circular chromosome. So for prokaryotes, it literally has to start in one spot, DNA B starts, and then it just goes all the way around until you've got the whole thing copied. And remember, prokaryotes do not do mitosis or meiosis, they do binary fission. Okay? For eukaryotes, linear chromosomes, it's got to start in multiple places. Every chromosome it's got to happen on, and it can happen in multiple places on those chromosomes. Okay, so that's one difference. All right, then we got into this. Okay? All right, so we know DNA codes for RNA. RNA gives you the information in order to put together a sequence of amino acids, a polypeptide, which can be folded into a correct protein, right? You can have, and now you should have the appreciation for it, you can have one, let's say, section of DNA right here, and you can make an mRNA copy of that DNA, but depending on how you splice and dice, your mature transcript could be very different, right? Exons and introns. You cut out the introns before it leaves the nucleus. what are her introns versus my introns versus your introns could be very, very different, right? And then that means what introns you cut out are going to vary the exons that are expressed. So you can have one gene coding for multiple polypeptide chains. Do you own that? Ask your bio buddy. Do you get that? Just based on processing. Okay? Now, a little reminder, RNA is single-stranded, whereas DNA is double-stranded. RNA has the base, what? Uracil, wherever you'd want to put a thymine. RNA has the sugar, ribose. DNA has the sugar, deoxyribose. They are the same language, though. What language are they? Nucleic acid. Does DNA become RNA? No. You use DNA to know how to construct your RNA. Okay, so don't ever say it becomes. You're using the DNA to construct your RNA. So I can just have I can have my nucleus that has my full house plans a la Winnie Lytton, right? All the house plans are in each cell, but maybe in my heart they pay attention to chapter 2, and in my liver they pay attention to chapter 4, and that's the mRNA they are making. Does that make sense? Okay. Now, mRNA is not the only RNA out there. What's another RNA? tRNA, which stands for transfer. What is its job to transfer? It has to get locked and loaded using ATP and an enzyme. The appropriate amino acid has to get put on the appropriate tRNA, yes? Okay, and if it has an amino acid on its head, what does it have at its feet? Anticodon, which is complementary to located on the mRNA. But that's not the only RNA. What other RNA do we have? rRNA, which stands for ribosomal. Ribosomes are made out of rRNA and protein. Remember when we studied the nucleus, there was a darker area right in the middle called that is where you're making rRNAs. Because you need a bunch of ribosomes, because it's ribosomes where you do protein synthesis. Okay? All right, now, we got that part down. So when we look at a picture like this does not scare us in any way, shape, or form. Because we see a portion of the DNA unwinding. We see us using the what strand, not the what strand. Leading strand, not the lagging strand. Okay? You're making a copy of mRNA right here. You're going to take this mRNA before, it says it's mature. So what has happened to it if it's mature? It's been put on a cap on the 5' end because you read from 3 to 5, making 5 to 3, so the head is now the 5' end. Put on a cap, 7-methylguanosine, and that's to what it? Protect it. And then you put on a poly-A tail. Poly-A means a bunch of nucleotides that all have a bunch of adenines in them. And it's about 100 to 200 nucleotides of adenine in it to protect the other end. And then you what? Cut out the, using spliceosomes, which are made out of snurps. I was slurping snurps. Obviously. I bet you did. All right, now. Do you remember that? I'll show you a picture. Remember spliceosomes where you cut out the introns, but they're made out of snurps. Once it's been spliced, capped and tailed, it can leave the nuclear envelope and it can go out into the cytoplasm and get on a workbench. What's our workbench called? But first the small ribosomal subunit, the mRNA, gets on there. The very first tRNA one is going to land in what spot? P spot. Always start in the P spot. Okay? In the P spot. And remember, the codon is going to be AUG. For every protein, first tRNA lands. If A would bind with an anticodon with U, AUU with A, G with C, that tRNA lands, and it's always carrying with it the very first amino acid, which is always what? Methionine. Say it. Methionine. Good. So then the large ribosomal subunit docks on, and now it's game on, right? tRNA2 lands. tRNA1 dumps his methionine onto tRNA2's amino acid, and he gets shifted into the exit spot, and the second tRNA gets shifted into the what spot? P spot. And another one lands in the A spot, and this is going to go on and on and on and on until you reach a stop codon. And that's what's going on right here. Okay? Now we can see this, here are the 3 RNAs, but we've already talked about this. The central dogma, right, says that you take the information in the DNA, and we already went over this, and you get a what copy of it? RNA copy. So it's nucleic acid to a different kind of nucleic acid, but it's still nucleic acid language. That's why that is called transcription, right? But then when you take that RNA and translate it into a phenotype, right, into some sort of protein, then that's when it's called translation. Just making more DNA, which was the first dance we did, because we're getting ready, we're in the S stage of the cell cycle, and we're going to get ready to do mitosis, right? That's when you do replication, DNA to more DNA. Did I lose you on anything? Okay, you feeling strong? Remember, this is universal, right? It's universal, and that leads to, my name is Luca. And remember, it's Nurnberg and Matai who came up with the genetic code. Remember, what do we look up? The codon, the anticodon, or the DNA? What do we look up? The codon. The mRNA, what does M stand for? Always look up the message. Okay, always look up the message. Every 3 bases are called a codon. So the process of forming the mRNA, remember, there's more to it than what I've given you, right? It's a little bit of a summary. But let's start right here. First, we've got to what? Unwind and unzip. Helicase, right? We have a leading strand and we have a lagging strand. Let's put the lagging strand away. Now, instead of using DNA polymerase, what are we going to use? RNA polymerase binds to the promoter. Say it again. RNA polymerase binds to the promoter. You're not going to start transcribing yet. This just docks us in. It's like a zipper on a jacket. You know, you got to feed it in, right? And then later you can move on. So it's just the feed-in spot is the promoter. We'll learn about the importance of the promoter tomorrow in class. One more time. RNA polymerase binds to the promoter. Boom, boom, boom. Where you have an A, you're bringing in a U nucleotide. G, C, T, A. Good. Make your mRNA. Peel it off. Set it aside. Go get that lagging strand. Bring it back. Wind it up. Put it away. Go get your mRNA. Time to process it. So we're going to put on a cap and a tail. And you cut out the introns before it leaves the nucleus. Now take your mRNA, go out and put it on a ribosome. First the small, and then the first tRNA lands, and then the large, right? Okay. Every 3 bases is called a codon, complementary to a anticodon. Start hooking the amino acids together. The ribosome does that. It facilitates forming what kind of bond? Peptide bond, polypeptides. Then you can fold it from its primary structure into a secondary and tertiary, possibly quaternary, for your fully functioning protein. How do you feel about that? You feel awesome. Okay? So remember, you can have exposed DNA, a leading strand, and RNA polymerase binds to the promoter. It can be boom booming, and here comes another one, boom booming, and here comes another one, boom booming. So boom, 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 all along here, lots of booming, okay? So you can have lots of transcripts out there. Same thing, when you have your mRNA sits on a ribosome, that ribosome can be moving right along. Here comes another one, here comes another one, here comes another one, here comes another one. What's significant is this. Remember, prokaryotic cells do not have a what? No nuclear envelope, right? They just have a nucleoid region. So, and they have no introns. They just have exons. They don't have that genetic junk. So the primary transcript can be getting formed. It is the transcript. It is mature from the time that it is formed. So you can literally be going boom, 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 making some mRNA, and it's dangling off. You're not even done. And a ribosome can get on it and start translating it immediately. Because it doesn't have to wait all that. Okay? It's good to go. you can start translating it immediately after you've transcribed it. Unlike eukaryotes, where we process it up and mature it up, and then we allow it to leave the nucleus, and then it goes out and gets on right its own. Ask your bio buddy, do you own that? Okay, and then I think, I talked to you about the chaperone proteins folding. That would be important. And then the DNA, you have coding and non-coding regions. Anything, if you code, the only part that's coding are mRNAs, right? Does that make sense to you? That's the only coding. tRNAs and rRNAs, that's non-coding portions of the DNA. Still critical, but you don't translate it into any kind of phenotype. And then if it gets screwed up, you have mutations. And then that is the end. And then this was one more picture I found, if this helps you. This has a good picture if you want to capture that, but it is in your presentation as well. Okay, you good? You feel strong? I am proud of you. Okay, make good choices. Have a piece of toast.